Assalamu alaikum. This is part 2 of the protein structure lecture. In this lecture, we'll be talking about tertiary structure as well as quaternary structure. So first, we talked about the primary structure, and we said that it is basically the sequence of amino acids that make up a polypeptide. And secondary structures are basically localized regional structures within a protein, and these are organized. So like alpha helices, turns, beta sheets, which are made of multiple beta strands, loops, coils, and so on. So these are defined structures within the protein itself. Now, tertiary structures are basically these are different definitions. So basically, we're talking about the overall conformation of a polypeptide chain. The three-dimensional structure, three-dimensional arrangement of all amino acid residues in space. The spatial arrangement of amino acids residues that are far apart in a sequence, in a primary structure sequence. So for example, this amino acid and this amino acid, they are located far away from each other in the primary structure, but they are close to each other in space. So basically, we're talking about the arrangement of amino acids in space. Okay. Now, there are different ways by which we can illustrate protein structures. We talked about the ribbon structure more than anything else. So basically, you have the alpha helices looking like ribbons, like helical rods, and the strands are illustrated as arrows, thick arrows. Okay, good. Where you note that the end of the arrow is basically the end of the beta strand, that is the one that is closer to the C-terminus of the polypeptide. Now, there are different ways to illustrate alpha helices, and that is you have this cylinder structure. Okay, beta strands still are illustrated as arrows. You can have a simple structure that looks like this, a trace structure, whereby you just draw the backbone of the polypeptide. You can have ball and stick structure, where atoms are small balls, but you have their orientation, their angular orientation, exactly as they look like in 3 dimensions. You can have something more complex that looks like this, a space-filling structure. Now here the balls are larger, okay, and you cannot see the backbone. Here with the ball and stick structure, you see the backbone, but with the space-filling structure, you don't see the backbone. Now you have something more modern, and this is computer made. It's known as the protein surface map. It shows the protein surface without the interior of the protein. Now, the protein surface map is important these days in designing drugs, because drugs would tend to bind to the surface of protein. So knowing how the surface of the protein looks like, that helps in drug design. So these are different ways to illustrate protein structure, tertiary structure of proteins. Now, the tertiary structure of proteins is determined by non-covalent interactions. Okay, not by the peptide bond, rather by non-covalent interactions. And here we're talking about the 4 non-covalent interactions, the 4 forces: the hydrogen bonds, the electrostatic interactions, the van der Waals interactions, and the hydrophobic interactions. So these are really the forces that determine how proteins look like. Right? So you have the hydrogen bonds within a protein. You can have a hydrophobic pocket. That is, you have a region within a protein that includes hydrophobic non-polar amino acids. You can have, again, you can have these hydrogen bonds within the peptide bond itself, the groups that make up the peptide bond. You can have what is known as salt bridge, and salt bridge is the same as electrostatic interactions. So if you have two atoms, for example, or two groups, and they are swimming in space, and they carry opposite charges, they would be attracted to each other via electrostatic interactions. But when we're talking about amino acid residues being part of a protein, and you have this electrostatic interaction, we call it a salt bridge. Okay. Again, you can have these hydrophobic interactions. You can also have a disulfide bond formed between two cysteine residues. Okay, and we'll talk about this as well. Now, so we have these non-covalent interactions, and these are important in determining protein structure. So hydrogen bonds, for example, occur not only within or between polypeptide chains, but also with the surrounding aqueous medium. Okay, that is where this protein is dissolved. Here we're talking about the solvent, like the water, the salt solution, a buffer, or whatever. So you can have this interaction again between amino acids and the surrounding molecules. Now you can have charge-charge interaction. These are salt bridges, as I said, not electrostatic interactions, but they're known as salt bridges. You can also have a charge dipole interaction. That is, you can have an interaction between a real charge with a partial positive or negative charge. Now, even within two oppositely charged amino acids like lysine, which is positively charged, the R group of lysine is positively charged, and glutamic acid or glutamate, and here you have a negative charge on the R group. Now you can have the formation of electrostatic interactions between these two amino acids, or you can have the formation of hydrogen bonds between the two groups as well. Okay, so these same charge groups, they can form either hydrogen bonds or electrostatic interactions. Van der Waals interactions are really important, even though they are the weakest forces, the weakest non-covalent forces, and they are the most transient, okay, because they keep on changing it. They depend on the position of electrons as electrons move around, move around within the atom itself. Okay, so at any certain fraction of a second, you have these electrons positioned opposite on one pole of an atom. So you have a partial positive charge right here, and you can have at the same time you can have these electrons clustered at one pole of another atom, and you can have this attraction between them. Okay, but this is all transient, and they change quickly. Or you can have repulsion as well. Okay, so you can have this attraction or repulsion, same thing. Now, I keep on telling the same story year after year after year, and that is, if you have a van der Waals interaction by itself, just one van der Waals interaction, its effect is negligible. It's nothing. It has no effect whatsoever. But because you have a large structure, large protein structure with a lot of van der Waals interactions, now these interactions do make a difference. So it's the same story of this father who gathered his 7 sons, and he gave each one of them a stick, and he asked them to break it, so each one broke his own stick. But then when he grouped all sticks together and he asked them to break him again, they couldn't. So having a lot of van der Waals interactions is really important in determining protein structure. Now, probably the most important force is actually hydrophobic interactions. The reason is that proteins having these nonpolar hydrophobic amino acids, now these amino acids, they're hydrophobic. They are afraid. They have this phobia from water. So as soon as a protein is formed, you have these hydrophobic amino acids clustering together, and they hide from water. They exist in the core of a protein. And on the surface of any protein, a globular rounded protein, you have these polar and charged amino acids. Okay, so probably the most important force that determines protein structure is basically hydrophobic interactions. Now, can polar and charged amino acids be found in the interior of proteins? And the answer is yes, they can be present. And they are present usually when you see a charged or a polar amino acid in the interior of a protein, you ask yourself immediately, what is the function, what is the purpose of this amino acid? Usually these amino acids that are charged, polar amino acids that are that reside, that are localized in the interior of proteins, they have a certain function. And we will learn about histidines being part of the function, the important function of hemoglobin and myoglobin. Now, there are stabilizing forces, and one of these forces is disulfide bonds, and another is the presence of a metal ion as part of a protein, that is a holo protein, right? So we have holo protein, apo protein. So when you have a non-protein molecule that is part of a protein, we call this protein a holo protein. Without this non-protein group, we call it an apo protein. So let's talk about disulfide bonds. Now, these are stabilizing forces. They are not structure determining forces. Okay, so the non-covalent interactions determine the protein three-dimensional structure. But what disulfide bonds do is that they stabilize the structure. Okay, so and these disulfide bonds are formed between two cysteine residues. So within a polypeptide, or let's say a polypeptide with another polypeptide forming one large protein multimeric. We'll talk about this later on. You can have the formation of disulfide bonds between these different polypeptides. Okay, linking polypeptides to each other, or within a protein, three-dimensional protein, you can have within the protein itself, the tertiary structure of this protein, you can have a disulfide bond. Okay, so so the disulfide bond is formed between two cysteine residues, forming a molecule or a group known as cysteine. So that is basically a result of oxidation. Now, when proteins are reduced, you break the disulfide bonds. Okay. Now, metal ions also play an important role in stabilizing the structure of proteins, and they also play a role in the function of these proteins as well. Now, these metal ions can be linked covalently or non-covalently with the amino acid residues. An example is carbonic anhydrase. This is an enzyme that contains zinc, and this zinc is bonded non-covalently to three histidine residues, stabilizing the structure of carbonic anhydrase, and it also plays an important role in the function of the enzyme. And you can have another protein that is myoglobin, and myoglobin contains a heme group. We'll talk about this later on. It contains a heme group, and this heme group is covalently linked to a histidine residue. Okay. Okay, remember we talked about supersecondary structures. And I said that supersecondary structures are basically a three-dimensional structure of a protein, a region of a protein, and it is composed of multiple secondary structures, combinations of alpha helices, beta strands, turns, loops, and whatever. Okay, now, so domains is another example of a supersecondary structure. So a domain is basically, again, it's a combination of alpha helices and beta strands or beta sheets, and they're connected to each other via turns and loops and whatever, and coils, and they are organized in a specific three-dimensional structure. Usually a domain may consist of 100 to 200 amino acid residues. What's really important here is that domains fold independently of the rest of the protein or of other domains within the same protein. So right here, you have this protein right here. This is one single protein. You can see that this in blue is actually part of a protein, and it forms a domain, a combination of multiple secondary structures. Now the thing is, if you cut this with scissors, for example, this would maintain its structure. This domain would maintain its structure, and it would also maintain its function, similar to this domain right here. Okay, so if you cut it from the other two domains, it can maintain its structure and it can also maintain its function, and same thing with this domain right here at the end. Now, similar domains can be found in proteins with similar functions and or structures, and they can also be present in different proteins. Okay, so the idea here is that domains can be associated with certain functions, like certain enzymatic activities or certain binding abilities, like you have DNA binding domains. Okay, the ability, so this domain is responsible for binding certain regions of DNA, and we talked about that in the molecular biology part of the course and in the previous course of molecular biology and cell biology. Now, something else is that when you have a domain or multiple domains within a protein, and these domains have a specific function, then they are known as folds. So, and they are given certain names. So for example, you can have what is known as the actin fold. You can have the nucleotide binding fold. So basically, this is a specified three-dimensional structure that is present in different proteins. And these proteins, even though overall they have different functions, they have this domain, and this domain has a similar purpose or similar function, like the nucleotide binding fold. So you can have different domains combined with each other, forming a nucleotide binding fold. Okay, like this one right here. Now the thing is, you can have this being present in different proteins, but these different proteins have different functions, but all of them can bind to a nucleotide like ATP, for example, because they have this domain, but they utilize ATP for different purposes. Now, something important about alpha helices is that they can form transmembrane domains. So you have membrane proteins that are inserted into plasma membranes. Now the part that goes into, that is embedded, that is buried in the plasma membrane has an alpha helical structure. Of course, you can imagine that the amino acids that make up the alpha helix are hydrophobic and non-polar, mainly hydrophobic and non-polar, so that there wouldn't be any repulsion between the fatty acids that make up the plasma membrane and the amino acids that make up the transmembrane domain. Now, if you have a protein with multiple transmembrane domains, these different alpha helices can be connected to each other via loops, turns, coils, whatever. Okay, and usually these turns and coils and loops, they have specific function. They are, for example, ligand binding sites. Ligand is basically a small molecule that binds to a receptor, okay, or to another molecule, to a protein, for example. Okay, like ATP, it can be a ligand, a growth hormone. It's a ligand for its receptor, for the growth hormone receptor, and so on. So usually these ligands bind to the amino acids that make up the loop structure of the protein. Now, something else about alpha helices is that you can have the formation of what is known as amphipathic alpha helices. Amphipathic means that it is a molecule or it is an object that has two opposite characteristics, that is hydrophilic and hydrophobic. Now, amphipathic alpha helices exist in channel proteins. So right here you have a channel, and this channel you have it has multiple transmembrane domains, and these transmembrane domains are connected to each other via, again, loops. Now, the purpose of this channel is to allow entry of ions, okay, and these ions are charged. Now the thing is, in order for ions to be attracted and to pass through the channel, the R groups of amino acids must be hydrophilic, okay, or oppositely charged, having a charge that is opposite to the charge of the ligand of the ion. Okay, so the R groups that make up the alpha helices must be again polar or charged. Now, on the other hand, the R groups that face the fatty acids, that face the plasma membrane, must be hydrophobic. So you have an alpha helix that is organized in a way where if you have a top view of the alpha helix, okay, so these are the amino acids that make up this alpha helix, you would find that on one side of the alpha helix you have the charged and polar amino acids, and this is the part that that faces the opening of the channel or the lumen of the channel. And if you look at the other side of the alpha helix, you can see that it is composed of mainly hydrophobic and non-polar amino acids, and this is the part that faces the fatty acids or the plasma membrane. So these are known as amphipathic alpha helices. All right, so now we talked about the four classes or the four levels of protein structure: primary, secondary, tertiary, and quaternary. So let's talk about properties of proteins right now. We'll specifically talk about denaturation and renaturation. We talked about denaturation in DNA before, right? So we said DNA denaturation is when you have the breaking of the non-covalent interactions, that is hydrogen bonds between the two strands. And renaturation is basically when the two strands recombine with each other, forming double-stranded DNA. Well, same thing with proteins. They can be denatured and they can be renatured as well. So denaturation is basically disruption of the native conformation, the three-dimensional structure of the protein, via breaking non-covalent bonds within the protein structure. Okay. Now, let's say that you have a protein that has disulfide linkages, as you can see in here, between these amino acids 40 and 95, and you have another one between amino acids 58 and 110, and so on. Now you can have complete disruption of such a protein by adding a reducing agent. So once you add a reducing agent like mercaptoethanol, beta-mercaptoethanol, the disulfide linkages are reduced, and you don't have disulfide bonds anymore. Now, there are a number of factors that do affect protein structure and cause protein denaturation. One of these factors is heat. What happens is that upon increasing temperatures, there would be a lot of kinetic energy in electrons, and that disrupts the van der Waals interactions between the different atoms, and that causes protein destabilization and protein denaturation. Something else is extreme pH. Since pH would affect electrostatic interactions or the salt bridges, and that can also result in also disrupting hydrogen bondings, and that would result in destabilization of protein structure, and it might lead to protein denaturation. Detergents can also cause protein denaturation simply because introducing a hydrophobic environment to proteins. That would causes the hydrophobic amino acids that are hidden uh, inside in the core of proteins to flip out and be exposed to the outside. Okay, so that again causes protein denaturation. One of these uh, detergents is known as Triton X100, which is non-ionic and it is uncharged, so it is fully hydrophobic. Another one is known as SDS, or sodium dodecyl sulfate. You'll learn about this SDS when we start talking, or when you learn about immunoblotting or Western blotting, which is similar to Southern that we talked about in the molecular biology course, except that it's for proteins. Now, SDS is an ionic detergent, so part of it is hydrophobic and another part is hydrophilic, and the hydrophobic part interacts with the hydrophobic interactions, and that results in protein denaturation. We have other chemicals like urea and guanidine hydrochloride. Now these result in disrupting hydrogen bonding and hydrophobic interactions, and they also result in protein denaturation. Now, in order to complete protein denaturation, we need to disrupt the disulfide bonds between the cysteines, and this happens by adding reducing agents, okay, like beta-mercaptoethanol and dithiothrietol. And what these do is that, again, they reduce the disulfide bonds, and that helps in protein denaturation. Again, disulfide bonds do not necessarily determine protein structure, but what they do is that they stabilize protein structure. So it's like putting a stick in a box. Okay, so you have an empty box, and this empty box you can push it left and right. You can wiggle it. Okay, it's not stable. But if you put a stick inside this box, um, this stick would stabilize the box itself, and you cannot wiggle it anymore. Okay, so it's something similar to that. You can. Okay. Now, if we remove the denaturing factors, proteins tend to renature. They tend to reform these non-covalent interactions. And this happens slowly. Okay, so and this was like an experiment that was done like in the 70s, and the scientist who did this experiment and explained it got the Nobel Prize for it. And basically, they had an enzyme, a ribonuclease enzyme, small enzyme. They added denaturing agents and the reducing factors, and then they started to remove these factors from proteins, and the protein itself, the enzyme, renatured, and it regained activity. Okay. And they realized in this experiment that that non-covalent interactions are the determining factors. So it's not really the disulfide bonds that determine protein structure. Now, so what are the main factors that determine protein structure? Basically, remember in the first slide, I said that you have 100 amino acid protein, so there are like gazillion possibilities of forming a protein structure, right? But this is not true, as I said, because it's basically the most stable structure would be formed, energetically speaking. So basically, proteins would use the least amount of energy needed to keep a protein stable. So there are really not gazillion possibilities, but very little probability of forming a certain protein structure. Okay. So what really determines protein structure is, again, the least amount of energy And this is dependent on the amino acid sequence. So remember this project that I told you about, the experiment that was done, or the prediction program that was developed to predict protein structure. It's huge development, huge advancement in protein biochemistry. So based on the amino acid sequence, you would be able to predict the best possible, energetically speaking, structure of a protein. So it's really the amino acid sequence, the primary structure. Okay, and this this energy and the amino acid sequence basically, um, is based on the proper angles of amino acids, where proteins, where amino acids would be placed in 3 dimensions and form the best non-covalent interactions, of course, starting with the hydrophobic interactions. Now, non-protein molecules also play a role, like I said, in stabilizing protein structure, like heme, like zinc, and other molecules. So it's easy for small proteins to renature without any help. But there are certain proteins, especially large ones, that these proteins need help. And help comes from other proteins known as chaperones. So, uh, and so what these chaperones do is that they look like barrels, okay? And there are like openings inside. So truly they look like barrels. And proteins would be inserted inside the chaperone proteins, and these chaperone proteins would start to fold proteins to to gain the proper structure of proteins. Now, so what happens is it's like folding a t-shirt, right? So it's easy, of course, to crumble your t-shirts and throw it on the bed. Well, that's the easy part. But so what's easier, or help is needed. So what chaperones do is that they take, like a shirt, they take this side of the shirt to the middle, and the other side to the middle again, and fold it up, and so on. So chaperones do exactly the same thing. They take proteins and they start putting groups together near each other, especially hydrophobic regions, because once a protein comes out, it is synthesized. Once it comes out of ribosomes, hydrophobic regions tend to associate with each other, trying to hide from water. Okay, so what chaperones do is that they take these proteins and they say, no, you hydrophobic region, I want you to be next to this hydrophobic region, not the one that appears after you. Okay, so that's the idea behind chaperones. And of course, energy is needed. So what chaperones do is that they form the proper structure of proteins, they fold the proteins properly by placing hydrophobic regions um, in the, again, the most energetically stabilizing form. So what happens is that proteins can misfold. That is, you can have proteins that are not properly formed, and the problem with that is that if you don't have this, is that proteins would tend to aggregate. Okay, and this aggregation is really a result of hydrophobic regions being exposed. So you have these hydrophobic regions of a protein that can aggregate with a hydrophobic region of another protein, and then a third protein comes in, and so on. So you can have this protein aggregation happening as a result of the interactions between the different hydrophobic regions of different proteins. And that causes a problem, because once you have a protein aggregate, you're talking about a large structure, and this structure would tend to precipitate, to accumulate in tissues, and that causes tissue damage. So this really happens in certain diseases. That is, you can have misfolded protein or partly folded proteins be aggregating in different tissues, and that causes certain diseases, as I will mention in a few seconds. So now these aggregates, they vary in size. You can have dimers, you can have trimers, you can have large protein structures, and these are collectively known as amyloids. Okay, um, so they can be toxic to cells. So an example is a disease known as a prion disease. Okay, so this prion disease is also known as Creutzfeldt-Jakob disease in humans. It's known as the mad cow disease in cows. It's known as scrapie in sheep. And I do remember, like in 20 years ago, there was a spread of mad cow disease in cows, in cattle, in England, and other countries, and that resulted in um, slaughtering of thousands of cattle in the UK. And the UK lost a lot in the cattle industry. So, and there was a huge controversy about importing cattle from the UK and other countries as well. So, and there was a fear that this prion, since it can be contagious, that this prion disease can be transferred from cattle to humans. So, and there were cases of that. So this prion disease is different than all other diseases. It is an infectious disease. Okay, it's different than other diseases in that it's not really caused by a virus. It is not caused by bacteria or any other pathogen. Rather, it is caused by a protein. Okay, and that was something really new to scientists in the 60s when they first discovered that, and there was a lot of um, argument about that and resistance against the idea that proteins can be infectious. Okay, and but it scientists discovered that there was a protein known as a prion protein, and this protein is misfolded and into an incorrect form, and it's known as a prion SC, and that resulted in the the aggregation of proteins. So what would happen is that a misfolded protein would bind to a correctly folded protein, and it causes the other protein, the correctly folded protein, to actually misfold. Okay, so that would lead to protein aggregation and the formation of this amyloid. Now, the correctly folded protein has alpha helices in its structure. It's mainly alpha helical. And the misfolded protein, you have the secondary structures change from alpha helices to strands and beta sheets. So the prion disease, it can be acquired by infection mainly. Now, it can also be inherited. So if the prion disease is mutated, that would result in increased probability of misfolding of the prion protein, or it can be spontaneous. Okay, just bad luck, let's say. So the disease is caused by a transmissible agent, and this transmissible agent is a protein. It is not, again, a pathogen of any type. Another disease that is caused by misfolded proteins is Alzheimer's disease, which is not transmissible among individuals. Okay, rather what you have is misfolding of proteins, a protein known as tau, or another protein known as the amyloid peptides. Okay, and that causes tissue damage, particularly in the brain, since these proteins exist in the brain. So what does cause Alzheimer's disease exactly? Now what happens is that you do have this amyloid protein on the cell surface, okay, and you have this transmembrane domain right here, okay, and normally what happens is that usually proteins on the cell surface do not stay there forever. Rather, you have what is known as protein shedding, okay, just like snakes changing their skin, so they shed their skin. Same idea. So every once in a while, what happens is that you have these enzymes, proteases, on the cell surface, and they cleave off proteins on the cell surface, and there's protein renewal. Okay, so normally you have this protein shedding. Now what happens is that there is an abnormality in protein shedding, resulting in the release of the hydrophobic region of the amyloid protein, and that results in aggregation of these hydrophobic regions. Okay, so this is one cause of Alzheimer's disease. Okay, so let's talk about quaternary structure now. Some proteins, not all proteins, some proteins have quaternary structure. These proteins are ones that are made of one polypeptide chain. Um, some are oligomeric. They have more than two polypeptide chains. Now, so basically a quaternary structure is basically the spatial arrangement of all polypeptide subunits and the nature of these interactions, of their interactions. Okay, so you have like, let's say a protein that is composed of three subunit polypeptides. You have alpha, beta, and gamma. Well, alpha is connected to beta, and beta is connected to gamma. So it's not like alpha and gamma are connected. So you're talking about the spatial arrangement of all of these subunits in space. Okay. So proteins can be composed of one subunit, and these are called monomers. They do not have quaternary structure. And you have other proteins that are made of two subunits, and these are known as dimers. Now, the simplest form of a protein with a quaternary structure is a homodimer, meaning that the two subunits are identical to each other. You can have proteins that are made of three subunits. These are trimers, four subunits, tetramers, and so on. So you can have proteins having as high as 60 subunits for certain enzymes like pyruvate dehydrogenase. Now, so each polypeptide chain is known as a subunit. Oligomeric proteins can be made of identical subunits, polypeptide chains, or different, and these are known as homo-oligomers or hetero-oligomers. Now, and usually when we say oligomer, we refer to proteins that have a quaternary structure and they have identical subunits. And when we say multimers, we are talking about proteins having different subunits. Now, the question is, how are these subunits connected to each other? Well, sometimes they can be connected via disulfide linkages, disulfide bonds, like immunoglobulins. Now, note that I keep on talking about, I try to give you similar examples in different lectures. So I talked about antibodies before. So and here I'm talking about antibodies as well. So you have these antibodies, which are also known as immunoglobulins. There are four polypeptides. You have what is known as the small chain, and you have the heavy chain, the light chain, and the heavy chain, and you have two of each. So you have two heavy chains and you have two light chains, and these chains are connected to each other via disulfide linkages, disulfide bonds. So they are linked to each other covalently. And we'll talk about hemoglobin, and hemoglobin is a tetramer protein. It's made of 2 subunits of alpha and 2 subunits of beta, and these subunits are linked to each other via non-covalent interaction. So the linkage of proteins, the way polypeptides are connected to each other in proteins having a quaternary structure, really depend on the proteins themselves. Now, so proteins can have simple structure, like just monomers with just tertiary structure, no quaternary structure, or you can have proteins with complex structures. Okay, not only that they are, not only that they have quaternary structure, but they can be associated with something else. So I did mention before that proteins can have non-protein groups associated with them, and we call these proteins conjugated proteins. Now these proteins, when they are covalently linked to a non-protein group, we call these proteins holoproteins. Okay, and the group, since it is covalently bonded to the protein, we call it a prosthetic group. Now, if we remove this non-protein group, then we call it an apoprotein. Okay, so whenever you hear the word apoprotein, you know that this is a protein that is normally associated with a non-protein group, but in this case, or we are talking about the protein without the non-protein group. If we say a holoprotein, we know that this is a protein that is associated covalently with a non-protein group. Okay? So it is a conjugated protein. Now, you can have different types of proteins, of such proteins. You can have lipoproteins, which are proteins associated with lipids. Okay, like you will learn like the lipoproteins that transport lipids in our body. Now, if I'm talking about the proteins themselves, I call them apolipoproteins. Okay, so these are the proteins that are normally associated with lipids, but I'm talking about the proteins themselves without the lipids. Phosphoproteins are proteins that are phosphorylated, or they can be phosphorylated. Hemoproteins are proteins associated with heme, like hemoglobin and myoglobin, and we'll talk about these two later on. Nucleoproteins are proteins that are associated with nucleic acids, and we have glycoproteins, which are proteins that are associated with carbohydrates. An example of proteins that are an example of glycoproteins is immunoglobulins again. Okay, so there are disulfide bonds, and they are proteins with a quaternary structure, but they are also proteins associated with carbohydrates. Now, how are these carbohydrates added to proteins? Well, there are two types of linkages. There is what we say N-linked sugars, and there are O-linked sugars. Now, N-linked sugars are sugars that are added to the R group of asparagine. Okay? And the O-linked sugars are sugars that are associated with the hydroxyl group of either serine or threonine. Now, when we start talking about collagen as an example of fibrous proteins, you will learn that sugars are associated with a modified lysine known as hydroxylysine.